को मैं साथ लेके जावांगा। मैं तेनू जिथे पावांगा मैं उत्थे बस जावांगा। मैं तेनू जिथे पावांगा मैं उत्थे बस जावांगा। मैं दिल दा हाल सुनावांगा। मैं तुझको ही यार बनावांगा। मैं अखियां मिलावांगा। अखियां मिलावांगा। मैं अखियां मिलावांगा। मैं यार बनावांगा। मैं
पवन माइक ऑन है।
रफ्ता रफ्ता सनम तुमसे मिली नजर तो हुआ है असर। ख्वाब सा ये जहां, फर्श धुआं धुआं, जन्नतों का शहर। हम तेरे हो गए या खुदा इश्क में गुमशुदा दिल जिगर। रफ्ता रफ्ता सनम तुमसे मिली नजर, तो हुआ है असर।
Yeah, good evening. Am I audible?
Good evening, sir.
Okay, why only 7 members? What happened to rest of the people?
Good evening, sir.
Yeah, good evening.
Good evening, sir.
Yeah, good evening. Good
How
evening
are
to
you, sir?
what happened to rest of the people? Yes.
Don't know, sir.
Hmm. Okay.
We don't know.
Oh, no, sir.
Okay, let us start. I will give one message in the group, then we will start. Try to revise meanwhile. We will give them, 2 minutes time to join.
Good evening, sir.
Sir, your voice is not coming.
सर ऑफलाइन क्लासेस कब से स्टार्ट होंगी?
Government has to decide. Hmm.
ओके सर.
Is it visible?
Yes, sir. It's visible.
Sir,
यस सर.
So in the previous session, we discussed the role of Stanley Cohen from Stanford University, along with Herbert Boyer, from the California University of San Francisco. Then here one person worked on the plasmids, Stanley Cohen worked on plasmids, then Herbert Boyer worked on this restriction enzymes.
Restriction enzyme, yes, sir.
Restriction enzymes. And here, you please remember how to remember. Generally, people will confuse. So, plasmids, you please put a code SP, Superintendent of Police. Stanley Cohen, plasmid. You remember like that, then the other person automatically, Herbert Boyer, who worked on restriction enzymes. Right? So, then here we also discussed what about the traditional view and what about the modern view in relation to this biotechnology. In the traditional view, the entire microorganism can be used in manufacturing of this curd and also bread, then wine, etc., etc. In the modern sense, we discussed that it is a genetically modified organism. The organism is genetically modified. Okay. And here we also discussed that if there is any reproductive problem, and that reproductive problem can be solved by having a test tube baby and by conducting in vitro fertilization. Okay. And this technology is helpful in this. And here we can also synthesize a gene, and you can use that by using this particular technology. And we can also prepare a DNA vaccine, and we can also see a defective gene. And here this is, we can correct this defective gene through this gene therapy. So this is what we discussed. And here, so we discussed the definition given by the European Federation of Biotechnology, and this European Federation of Biotechnology defined this biotechnology as the integration of the natural science with the organisms, maybe with the organisms, maybe with the cells, maybe with these parts of the cells, maybe with the molecular analogs. And here thereby we can produce and we can do services even. So here there is an integration between the natural science and organisms as a whole, or one second, just my commissioner is calling. see here. Here in relation to this, we need to know what are the different tools which are there in this biotechnology. See. Yeah. See here. In the case of this biotechnology, we discussed that there are two important core techniques that are responsible for this modern biotechnology. See, traditionally, if you see, because here these bits are important. Traditionally, if you see, in the case of asexual reproduction, so what about the asexual reproduction? There is no question of any sexual, mating and other process, because this is asexual. The information which is present in the parents will be transmitted as it is. As it is transmitting as it is. That means here there is no much difference between the parents and the next progeny. So what? Yes.
Sir, the one who has joined very late the classes, for them, how they will get the recorded lectures? The previous thing.
Yeah, previous lectures. Yes, you can get the
I have
lecture.
mailed it many times, sir, but still there is no response.
Yeah, you can message me. My number is there in the group.
Yes,
You
sir.
can. I will forward to the concerned people. And
Okay,
I will-
sir. Okay, sir.
Right, Amrit.
Thank you, sir.
See here. In the case of asexual reproduction, wherein whatever the information that is present in the parents will be transmitted to the progeny. That means here the information which is present in the parents will be transmitted. So the genetic information which is present here in the parents will be transmitted. It is preserving. So in the asexual reproduction, it preserves the genetic information. That means no variation. This is what you should remember. As a result of reproduction, there is no variation. Variation will not be seen. Whatever the parental genetic information is present as it is, it will be, spread. In the case of sexual reproduction, we discussed, meiosis process. Meiosis in male, meiosis in female, exhaustively we have discussed. And here the meiosis in male and female will produce what? Will produce variation. We discussed, how they, parental genetic material and maternal genetic material, will combine in the process of, prophase 1. We discussed the prophase 1. What about prophase 1? Prophase 1 can be discussed under how many heads? We discussed. How many heads? Yes, please.
So 5.
Yeah, 5. Leptotin,
Leptotin, zygotin, pectine.
diplotin, diakinesis. And here in leptotin, what we discussed? In leptotin, the homology such will occur. And here, what is homology such? Their parental, paternal and maternal chromosomes will try to identify each other. And here, this is the paternal chromosome, and here this is the maternal chromosome, and they will come together by the time of the zygote. So 2 are present, and hence they are otherwise called as bivalents. Bivalents. And how many such bivalents are present in the cell? This is one chromosome, and this
23.
is other. Yeah, 23 bivalents are present. And slowly over a period of time, what will happen? From the leptotin through the zygotin and pachytine, condensation, coiling, dehydration occurs, and thereby this single line will appear as a chromosome, and this single line will also appear as a chromosome. So one is father chromosome, another one is mother chromosome. All these things may occur, say maybe in the case of, male individual. Male will have his father and his mother. And the same thing may occur in the, female. And female will also have her mother and her father. So here, neither in the male or female, their father chromosome and their mother chromosome will come together and will follow condensation, coiling and dehydration, and thereby the details will be seen. This is the parental chromosome, father chromosome. This is mother chromosome. And here they will exchange the genetic material. And once they exchange the genetic material, that process is what is called as the crossing over. Exchange of genetic material among the non-sister chromatids of the homologous pair. That is what we discussed. So what will happen here? This genetic material, which is having mother, will also have father material because of the crossing over. So it is not a pure one. One chromatid, this is one chromatid, it will also have, other genetic material like this, impurity in one chromatid. So one is impure one, another one is a pure one. And this, in anaphase, what will happen? Prophase 1, metaphase 1, anaphase 1, then telophase. In anaphase, what is the characteristic feature? And
Sir,
here, this
homologous
chromatid will
chromosome separate.
go to one gamete and this chromatid will go to another gamete. So two gametes are produced here. And from other one, other two gametes are produced. So one gametocyte will produce 4 varieties of cells. 4 are different. Each one is different. That means here the parent is not transmitting the as it is genetic information to the next generation. So here the parent is producing variation. That is possible only in the case of meiosis. So this, reproductive, mechanism is producing. The sexual reproduction is producing variation. Number of variations are produced. Not one variety. One spermatozoa will have one variety of chromosome. Another spermatozoa will have other variety. Another spermatozoa will have some other variety. And some other spermatozoa will have some other variety. So no two spermatozoa are same. No two spermatozoa are same. Is it clear?
Yes, sir.
And hence, what is the most important point? What about the scenario in the case of asexual reproduction? All this business will not occur. This entire business will not occur. So what will occur? Asexual reproduction occurs, no variation. So when there is sexual reproduction, you should remember that there will be variation. That is what you should remember. Is it clear?
Yes, sir.
So in the case of asexual reproduction, there is variation. Asexual reproduction, there is no variation. The genetic information will be preserved. Preservation of genetic material. In the case of sexual reproduction, preservation is not possible because recombination occurs, because the crossing over occurs, and hence, different varieties of gametes are produced. So sexual reproduction is providing opportunities for the variations and formulation of unique combination of genetic setup. And some of the setups may be beneficial to the organism as well as to the population. It is beneficial to the organism and hence it is important for the population. Then, if you take up the other area. What is this other area? The traditional hybridization procedures. We discussed the Mendel's laws. In the case of this Mendel's laws, Mendel conducted in the case of hybridization, Mendel conducted the hybridization experiments. What about hybridization? Not only Mendel conducted on the plants, plant hybridization, plant breeding otherwise, plant breeding experiments. Not only plant breeding experiments, animal breeding experiments even. You might have seen dogs, and one dog will have one character, other dog will have other character. And if you mate with these two dogs, then what will happen? The characters present in one particular dog and the characters present in other dogs also enter into the next generation. Not only that, here the unwanted characters will also enter into the next generation. Because you are dealing with the entire organism as a whole. You are mating with one animal with the other animal and you are producing a new one. The new one is what is called as hybrid. Okay. In the case of Mendel's experiments, we discussed, hybrids, F1 generation, F2 generation, F3 generation, like that. So what is the issue with the hybrids? There are some best quality genes in one particular either plant or animal. And here these best quality genes will be transmitted apart from the best quality genes. If worst genes, if disadvantaged genes are present, they will also get transmitted to the next generation. That means here, multiplication of undesirable genes along with the desirable genes will also occur. Okay. When? Very often.
Yes, sir.
Very often in the case of Yogita, you please remove that cat symbol. And I am afraid of by seeing that symbol.
Okay, sir.
Kore, my dear, you please use your photo. That is a beautiful photo. Why should you use this nonsense photo? Huh?
Okay, sir.
Right. Good. Yeah. Here, very often, multiplication of undesirable genes along with the desirable genes will occur. And how to delete these desirable, undesirable genes? Undesirable genes, nobody want undesirable genes. But here they will enter. How to prevent? You cannot prevent. That is the problem with the traditional hybridization procedures. In the case of traditional hybridization techniques, very often the undesirable genes will be included. That is what is important. Undesirable genes. This is the important point. So how to avoid this? The alternative is this one, recombinant DNA technology. And here if you see the entire genetic engineering, this is nothing but the production of the recombinant DNA, then gene cloning, then gene transfer. That is nothing but only which gene will be transferred, which gene will be cloned. And here the gene that is cloned means multiplied, which will be multiplied and which will be transferred, which will be recombined with the other source for the multiplication, for the
desirable gene.
Only desirable. And we will not touch this undesirable activity. Undesirable will not be transmitted to the next generation. So what is the best alternative to the traditional hybridization techniques and which are having this disadvantage of including these undesirable genes into the next generation? Genetic engineering. That is nothing but the recombinant DNA technology. That is nothing but the cloning mechanism. That is nothing but the gene transfer to the next generations. Is it clear?
Yes, sir.
Yeah. There are certain issues and problems in the hybridization mechanisms. When these hybridization mechanisms were introduced, since Mesolithic age, man started interfering in the plant and also animal. Especially it started with the dog, then later on goat, later on sheep, later on cattle, camel, ass, in different pockets of the world, different animals, as per their wish, they started combining. They started mating one animal with the other animal. Then started producing new varieties. That is how, see the dog breeds, numerous varieties of dog breeds are there. Along with the desirable character, undesirable character will also be entered into the next generation. That can be removed. The undesirable genes can be removed only by following this genetic engineering mechanism. That is what, okay? So
Yes,
here,
sir.
what is the importance, what is the significance of this recombinant DNA technology? If there is any question on significance of this recombinant DNA technology for your subjective question, what is the significance? The significance is there is a limitation in the case of traditional hybridization techniques with respect to the plant breeding, with respect to the animal breeding, and this limitation can be overcome. How? Because this recombinant DNA technology is allowing us to isolate and introduce only desirable genes. It may be one gene or a set of genes. Okay, without this, out is important, without introducing undesirable genes into the target organism. What is the target organism? We discussed that this is the bacteria. And here we are removing this circular DNA from the bacteria. And here this is the plasmid, because this plasmid is present in bacteria. And here this is the human DNA. And there is desirable DNA here. This desirable DNA is this one. And this can be introduced into this plasmid. And again, this plasmid can be introduced into the bacterium. This is what is the target organism. Because this target organism, that is nothing but bacteria, will produce the concerned gene product. If this gene is producing insulin, yes, this bacteria will now produce insulin. If this gene is producing factor 8, yes, now bacteria will produce factor 8. Okay, if this gene is involved in the production of growth hormone, yes, now this bacteria, which is the target organism, will produce growth hormone. Where? In the bioreactor. What is reactor and other things we will see. And only thing is, you need to provide some material for the multiplication, for the asexual reproduction of this bacteria, because you know that asexual reproduction occurs in this bacteria, which is otherwise called as amitosis. Amitosis occurs, and thereby one bacteria, whatever that is there, so if this gene is present, yes, this gene will be passed on to the next one without any variation. If there is any variation, your product will not be produced. That is why I have explained asexual reproduction, it will not produce variation. If it is a sexually reproducing organism, why we are not using other cells, other organisms, other life forms for the production of these different proteins? Because sexual reproduction is essential. Variation should not be there. Suppose this is the insulin gene, and here insulin gene should be present, and it should not pass on, because this bacteria will divide through the asexual, through the amitosis. Are you able to follow?
Yes, sir.
If sexual reproduction occurs, what will happen? There is every possibility of mixing this particular gene with some other material, and thereby the next generation bacteria will not have the same gene, and hence you cannot produce the same insulin. For that particular purpose, the target organism always will be this bacteria. That is why in the bioreactors, this bacteria will be taken and bacteria should multiply. For the multiplication, yes, we have to add medium. What is that medium? That act as nutrients. Medium is nothing but a combination of different nutrients. And all these nutrients will be taken out by this bacteria and will multiply. And hence, large sized bioreactors can be used in the biotechnology industry, in the pharmaceutical industry. Is it clear? So, there is no question of
Yes, sir.
introduction of undesirable genes into the target organisms. Only the desirable genes will be introduced, not the undesirable genes into the target organism. The target organism is the bacteria. Okay, and this is how that there is a problem, there is a limitation in the hybridization technique. And that limitation is nothing but introduction of the undesirable genes that will be avoided by this recombinant DNA technology or gene cloning mechanism or gene transferring mechanism. Any questions?
No, sir.
Yeah. See here, what is there in the, what are the different keywords in the genetic engineering process? What is there in the genetic engineering process? We will see the process and come back to this again. First, we will read what is here, then we will see the process and come to this. Techniques to alter the chemistry of genetic material. Genetic material may be DNA, maybe RNA. To introduce these, DNA or RNA into the host organisms, and thereby the host will change. Host will change means the expression of the host will change. Just now we discussed this bacteria, which is having a gene responsible for insulin. Normally, insulin will not be produced by the bacteria. Now the phenotype means the expression of the bacteria will change. Bacteria will now started producing insulin. Why? The DNA or RNA can be introduced into this. And thereby the phenotype of the organism will change. Now this is the host. Host is taking up the plasmid with this desirable DNA. Then, and where this host can be grown, and there must be a sterile environment. Sterile environment means other microorganisms should not enter into this. That means contamination free. What contamination free? Other microbes should not be present. And here the maintenance of sterile ambience, that is the microbial contamination free ambience, that is possible only through this chemical engineering process. That means without this chemical engineering process, this genetic engineering is not possible. And why this chemical engineering process? To enable growth of only the desired microbe. Large quantities, not small quantities. Very large quantities must be produced. So the desired microbe growth is essential. And this is possible only in this contamination free. If other microbiome is present, if other bacteria is present, if other organisms are present, they will also consume the nutrients which are present in the bioreactor. And they will also grow. And here our interest is our desired microbes should grow. Our desired microbes should grow, not other microbe. So our interest is only the desired microbe. What is the desired microbe? That is what you need to understand. Only the microbe which is essential.
Sir,
you are
please
not
unmute
audible.
your mic.
Yeah. See, why this desired microbe should grow? What is the purpose? The purpose is because the desired microbe is having the desired gene and that must be produced. So for the manufacturing of biotechnological products, what are those products? It may be antibiotics, it may be vaccines, it may be enzymes. These are all the different products which are produced. So what you should do now? The technique we need to take up, we need to alter. What you should alter? You should alter the plasmid, chemistry. The chemistry of the genetic material must be altered. So normally this is the plasmid and you need to add one more. You need to add one more gene. You need to alter the genetic material. You can alter. And this altered genetic material can be introduced into the host. What is the host? Bacteria. Thereby, bacteria will change its phenotype. And only this particular desired microbe should grow and not the other microbe. And hence, microbial contamination-free ambience must be maintained. And that is possible through this chemical engineering process. And here, through this, the desired microbe should grow in large quantities. For what purpose? For the production of enzymes, for the production of vaccines, for the production of antibiotics, and for the production of other proteins. This is what is genetic engineering process. Is it clear?
Yes, sir.
Yes,
Yes,
sir.
sir.
Now we will see what exactly is the process. This is the same thing can be discussed in other way. And how it will be produced and how the host, genetic material phenotype can be changed, etc., etc. We need to see. Okay. Yeah, see this. This is the process of recombinant DNA technology. So we discussed that. This is the human genome. And there is our desired DNA. There is desired DNA. This desired DNA is nothing but desired gene. Because gene is nothing but segment of the DNA. And here, by utilizing a restriction enzyme, we discussed how the restriction enzyme will cut the DNA. And thereby, yes, now the desired DNA is here. Desired DNA is here. And there is a bacteria. And in the bacteria, there is plasmid. And this is the bacterial chromosome. And this plasmid is the self-replicating material we discussed, which is circular in nature, which is double stranded. These are the three characters. The plasmid will have self-replication mechanism. The plasmid is double stranded. The plasmid is circular. This is extra chromosomal material. These are the things. Plasmids studied by who? Just now we discussed SP. Who studied?
Stanley Cohen.
Stanley Cohen. Stanley Cohen. And who is the other person who worked on restriction enzymes?
Sir Herbert Boyer.
Herbert Boyer. Okay, fine, good. Now we need to isolate this plasmid. Now isolated. Okay. You have to use the same restriction enzyme on this so that you know a portion will be cut down. As a result of this will happen. Now you can combine these two. This is the DNA from one source, human source, and this is the DNA from bacteria source. We are recombining these two sources, and hence now the recombinant DNA can be seen. So what is that recombinant DNA? This is recombinant DNA. This is recombinant rDNA. This is the process. And this rDNA, this is nothing. We are engineering the DNA from two different sources. That is why this is what is called as genetic engineering. We are isolating the DNA segments. And now what you can do, you can introduce this into bacteria. This is the host. And what is the capacity of this plasmid? And this capacity of the plasmid is, it can enter into any cell, including bacterial cell. That is the capacity. It can enter. And there are methods, we will see those methods. How to push this, how to prepare this, and how to prepare this host. And thereby, this particular plasmid can be introduced into this host. And there are a number of methods to introduce the plasmid into the host. We will see all those methods and how the plasmid can be introduced into the host. Right? See, finally, this is what is host, and this host should be multiplied. How to multiply? For that particular purpose, we will use bioreactor, and this bioreactor will have nutrition medium. Nutrition will be provided, and we discuss the nutritional control of growth and development while discussing mitosis. And there is one critical ratio between the volume of the cell and the cell surface. When you take the nutrition, then the volume will increase. And here, when the volume will increase, the surface area should also increase to maintain this critical ratio. Otherwise, the cell will die. That is what we discussed. When the volume increases, the surface area should increase. And the best way to increase the surface area is division. And hence, this bacteria will divide. And automatically, the plasmid which is present here in this bacteria will also be passed on to the next generation. And thereby, millions and millions of bacteria will be multiplied in this bioreactor. That is the purpose of this bioreactor. And the bioreactor will have the bacteria which is having a recombinant DNA. And this is what is called as transformed bacteria. Transformant otherwise. And other bacteria which are not having this recombinant plasmid are non-transformants. Non-transformants. Suppose if you take the test tube and if you keep this recombinant DNA into this test tube, and here several bacteria are present, and some are non-transformants and some are transformants, and we need to identify only the transformants, because into the bioreactor, and we need to multiply only this category, not this category. For that particular purpose, we are using different biomarkers. And how to detect the bacteria, or how to detect this transformant which is having this recombinant DNA is a real challenge. And here we can identify very easily the biomarkers. By using the biomarkers, some of the antibiotic resistance genes and some other varieties of markers are used in the biotechnology industry. Okay, and by using those biomarkers, yes, we can identify easily the transformants and we can grow them in the bioreactor. And you need not grow, these are useless. And they will take the nutrition and it is very costly. Giving this nutrition in the bioreactor is a costly method. So you should not waste your nutrition on the useless non-transferments. So if you able to identify the transferment bacteria and you can give this nutrition only to these transferments, not this non-transferments. No to this, no nutrition should be given. So what is the most important critical factor? Identification of these transferments, which are nothing but the bacteria which are having this recombinant DNA, recombinant plasmid otherwise. So what you are using? By using the biomarkers. Is it clear?
Yes, sir.
Yes, sir.
Yes, sir.
Yes,
Only
sir.
those transformants can be grown in the bioreactor in a very big way, in the largest scale possible. Okay, then you see this.
Sir,
Yes.
sir, why we are using same restriction enzyme to cut the plasmid as well as desired DNA?
Otherwise, sticky ends are not same. To have the same sticky ends, because the attachment is a difficult issue, and hence we need to use the same restriction enzyme to have the same sticky ends. This is bit. Is it clear? How we will produce sticky ends, we will see in the restriction enzymes. I will
Okay,
explain.
Okay,
For
sir.
the time being, you remember this. And I will give the fill in the blank type of questions wherein this bit will be given to produce the same sticky ends. And the same technology is helpful in the DNA fingerprinting technology given by Alex Jeffries in the year 1984. Is it clear? Are you getting the idea on DNA fingerprinting technology and what we discussed in DNA fingerprinting technology?
Yes, sir.
Yes. Now you see. You see now.
So what happened in 1984?
What happened? You tell me. We discussed. What happened? Alex Jeffries, DNA fingerprinting, DNA probe we discussed, restriction enzymes we discussed, and the same restriction enzyme should be used on the sample DNA and also the suspected individuals. On all the suspected individuals, same restriction enzyme you need to use. And if you change the restriction enzyme, then the result will change and the identification will become very difficult task. Discussed or not discussed?
Sir, discussed.
Yes, sir, discussed.
You see now, see what we are discussing is the process of, rDNA technology. In the process of rDNA technology, see, familiarize with these, diagrammatic representations. Once we are, you are acquainted with this, diagrams, then it will be very easy just before the day of examination to revise. And a number of such connections will be given. So the entire thing, biotechnology can be discussed in 3-4 sheets, not more than that. Only thing is, try to get familiarity with these things so that the revision will be very easy before the day of examination. And here we will give these printouts to you in the color format. Don't worry. So once this process will start, once you receive this stipend, then you will also receive these things in the color format. See, this is, process of rDNA technology. That is what I have discussed and put it in this form. See here. First one is, you need to isolate the genetic material. How to isolate the genetic material is the real challenge. How to isolate? We have discussed, very easily that this is the, human genome, and from this, yes, from this there is desired DNA, and this can be isolated. How to isolate? Then we also discussed that this restriction enzyme can be digested, and this is not easy one. Where this human genome is present? Within the nuclear envelope. Is it freely available? No, it is there in the
No,
nucleus.
sir.
And here, this is the cellular envelope. This is what is called as the plasma membrane. And this is what is called as a cytoplasm. And this is what is called as a nucleoplasm. And these two can be combinedly called as a protoplasm. Okay. And in cytoplasm, number of organelles are present. Mitochondria is present. Then, endoplasmic reticulum is present. Lysozymes are present. Centrioles are present in the centrosome. Different cell organelles are present. And here you have to detach this plasma membrane. You have to remove all these. And finally, you need to go to this, cytoplasm, nuclear envelope. Then this genetic material is present. So here, what is this genetic material? This is what is human genome. In the evolution, I have discussed in my initial sessions, what we discussed. See, on the earth, first RNA. Okay, first RNA. And after RNA, evolution, slowly DNA evolved. RNA is the single stranded one. DNA is the double stranded one. Then slowly this double stranded combines with histone proteins, non-histone proteins. Okay, and here combined with histone proteins and non-histone proteins, because this DNA is negatively charged one or positively charged one?
Sir, negatively.
Negative.
DNA is negative. These histone proteins are positive, and hence, the negatively charged DNA wrapped around the histone proteins, positive charges, and thereby coiling and condensation occurs, and from this, the chromatid, the chromosome will be formed. That means here RNA evolution, then later on DNA evolution, later on combination with histone and non-histone protein, and finally chromosome is formed. Now we want this DNA, segment of the DNA. So what you should delete? You have to delete these histone proteins and non-histone proteins from this material. And not only that, this RNA is also combined with this DNA. Because some of these materials are present. You might have studied nucleolus. What is that nucleolus? Nuclear organizing region, NOR. This will produce RNA. And you know that RNA are of three kinds: rRNA, ribosomal RNA, tRNA, transfer RNA, RNA, messenger RNA. And all these RNAs are produced by this nucleolus. Nucleolus is nothing but a nuclear organizing region. Where is the location of the nuclear organizing region? And we studied that in the case of mitosis. Mitosis can be discussed under prophase, metaphase, anaphase, telophase. In the prophase, what we discussed? By the end of the prophase,
proteins
the envelope will be deleted. Isn't it? Nucleolus and nuclear envelope will be deleted. So when there is a network, nucleolus will be seen. This is what we discussed. What is the purpose of this nuclear organizing region? It will produce different varieties of rRNA. So rRNA is also present here. So RNA is present here. Proteins are present here. Some of the proteins are having histones, and some of the proteins are having non-histone amino acids. And all these different varieties of substances are present. And all these substances must be deleted. Then only the restriction enzyme will act on this pure DNA. So under this backdrop, now you read all these things. This is the backdrop. Now you read this. So DNA, cutting of DNA with restriction enzyme is not so simple. I have simplified those things. It is not so simple. So you have to remove the different substances. Now, how to isolate the genetic material, that is DNA, desired DNA? See this. Nucleic acid is the genetic material of all the organisms without any exception. Is there any problem in this sentence? Nucleic acid is the genetic material. What nucleic acid? It may be RNA or DNA. In majority of the organisms, deoxyribonucleic acid is the genetic material. DNA is the genetic material in majority. So what about the genes? Genes are located on the long molecules of DNA. Okay, and these are intertwined with proteins and histones. Proteins like, the histone proteins and non-histone proteins. In order to cut the DNA with restriction enzymes, what is essential? You should take it needs to be in the pure form. What is the pure form? No RNA, no protein, maybe histone or non-histone, nothing. So, okay, since the DNA is enclosed within the membranes, we have to break the cell open to release the DNA along with macromolecules such as RNA, proteins, polysaccharides, lipids, all these substances. Are you able to follow?
Yes, sir.
Okay, that means here, in order to cut the DNA from the restriction with the restriction enzymes, other macromolecules should not be present. Other macromolecules should not be present. What are these macromolecules? Yes, these are the macromolecules. Proteins are there, RNA is there, polysaccharide is there, lipid is there. All these things must be detached. Okay.
Yes, sir.
Okay, how to achieve these things? This is fine. How to achieve? How to keep this in the purest form? This can be done by, this can be achieved by treating the bacterial cells or plant cells or animal cells with, enzymes. What are those enzymes? For the bacteria, lysozyme can be used. For the plant cells, cellulose can be used because the plant cells will have, the cell wall. Cell wall consists of cellulose, hemicellulose, and pectin that must be digested to enter into. Fungus will have chitinous material. So different enzymes for different cells can be used to get this purest form. Not only that. How to remove the proteins? How to remove the proteins? You see, the proteins can be removed by protease. The proteins can be removed by protease. And other molecules are also there. Appropriate treatment. After removal of all macromolecules, add chilled ethanol. You have to add chilled ethanol. This is bit. Ethanol. You should add ethanol. Okay. And here, purified DNA ultimately precipitates. Okay. And how it will be seen? It will be seen as fine threads in the suspension. Then you can take out by the method called spooling. Okay, spooling. See this? See this? It will be appeared like this. So how to remove this RNA? RNA can be removed by ribonuclease. Proteins can be removed by protease, and others can be used. And purest form, it will appear like threads. And separated DNA can be by spooling. You can separate the DNA. So this is the pure DNA without any protein, RNA, and other molecules. Is it clear? That is how we will isolate this pure DNA. First, this is the first step in the recombinant DNA technology. So isolation of the DNA is the first step. And how to isolate? You have to remove all the other substances. Proteins can be removed by protease. Then ribonuclease can be used to remove this ribonucleic acid. And for the bacterial cells, you have to use, lysosomes. And for the plant cells, you need to use cellulase. And for the fungus, you need to use chitinase, like this. So how to remove this, thread-like pure DNA? By the process called spooling. Right. This is the first step. Then what is the next step? Now, this is the DNA. This is the DNA, pure DNA. You have to cut the DNA at specific locations. At only specific locations. Who will cut? Restriction enzyme digestion. You need to add restriction enzyme to this isolated DNA by above method, mentioned method. Okay. See here. What we discussed? Use the same restriction enzyme, same specific restriction enzyme on both source DNA as well as vector DNA. What is this vector DNA? One is source DNA. What is the source DNA? Source DNA is nothing but source for insulin gene, source for growth hormone gene, source for factor 8, and source for factor 9. This is source. Where is that source? Where all these substances are essential? In human being. So you need to take human source. And this is the source DNA. And you have to cut. Then what about this vector? Somebody else must transmit. You have studied, vector-borne diseases. What are those vector-borne diseases? You have studied vector-borne diseases. What are those vector-borne diseases? Suppose, say, for example, Plasmodium, carried by, female. Which mosquito? Which mosquito? Female Anopheles mosquito. That is a vector. Female Anopheles will act as vector. Elephantiasis transmitted by Culex, vector. And here, who is carrying this desired DNA? Plasmid is carrying. That is what is vector DNA. So, what this sentence is giving? Same restriction enzyme can be used and source can be used and vector DNA. Is it clear?
Yes, sir.
Yeah. So read this one. So by giving the optimal conditions, then incubate the purified DNA molecules with the restriction enzyme at optimal conditions for that specific enzyme activity. Then, and this agarose gel electrophoresis is used. This is simply called as gel electrophoresis. We will discuss what is this gel electrophoresis and how it is used. For the time being, you remember this. We have discussed previously this agarose gel electrophoresis. Okay, and this is employed to see the progression of this restriction enzyme digestion. And here, DNA, you know that just now we discussed, DNA is the negatively charged one. And hence, it moves towards the positively charged one, that is nothing but the anode. And here, the DNA in the gel electrophoresis will have one positive electrode, another one is a negative electrode, and this negatively charged one will move towards the positively charged anode. You know, anode is positively charged. Okay, and here the same process of gel electrophoresis can be conducted for vector DNA also, so that you can cut this restriction enzyme and you can use this restriction enzyme on the vector DNA as well as source DNA. Then, just now we discussed, these two must be combined. So preparation of this recombinant DNA by adding ligase. So you have to add ligase so that recombinant DNA can be seen. So what you should do, we discussed in vivo and in vitro fertilization, in vitro gene cloning. That in vitro gene cloning is through.
सर इनके आपने माइंड मैप्स ग्रुप पे नहीं डाले ना?
Yeah.
सर माइंड मैप्स ग्रुप पे नहीं भेजे थे ना आपने?
Yeah, we will. Yes.
Okay, sir.
Yeah, in vitro is nothing but polymerization chain reaction. We discussed this. I have discussed the mechanism of PCR and how it will work. Discussed or not discussed?
Yes, sir, discussed.
Yes, sir, discussed.
Discussed. Yes, sir.
Yeah, in vitro method to produce multiple copies of the desired gene. So number of copies of gene of interest. So this is our gene of interest. Say for example, this is our gene of interest. And this is 5 prime, and this is 3 prime, 3 prime, 5 prime. And there are 3 steps in this. One is the denaturation. In the denaturation, you have to increase the heat so that the 2 strands will separate. And here, there will be a primer here. And there will be a primer here. And this will be extended like this. And this will be extended like this. And thereby, this will become a new DNA. And this will become a new DNA. It will be extended. So, denaturation, annealing, and extension. And here we also discussed that there is polymerization which is essential here because this PCR needs 4 nucleotides, 4 varieties of nucleotides. And here all these 4 nucleotides will be added. That process is what is called as a polymerization. And here to add this polymerization, for the polymerization process, there is one enzyme called as DNA polymerase. But the DNA polymerase will be deactivated with increase in temperature. That is possible in the case of denaturation. So for each and every cycle, you need to add this polymerase. And that was removed by the isolation of Taq polymerase. From the Thermus aquaticus bacterium, the polymerase enzyme was removed, and that is the thermostable, temperature resistant. And if you add this, you need not stop the PCR mission for each and every cycle. So after 30 cycles, 1 billion times the same DNA will be multiplied. That is the capacity of this PCR machine. So each cycle has how many steps? Each cycle has how many steps? 3 steps. Denaturation, primer annealing, then extension of the primers. These are the
3
3
steps.
Yes.
Denaturation, annealing.
And extension of the primers.
Sir, what we do in annealing?
Annealing means the 2 strands will come together. The old strand and the new strand will come together. Okay. See, this is
Sir, why they come together?
if you reduce the temperature, then they will come together. Because
We separate them in denaturation. Why they will come together?
complementary base pairing mechanism will work out. Then the hydrogen bonds will be formed. Then the 2 strands will come together. And here the temperature must be decreased. And this primer should be annealed, should be linked with this old strand. That is why it is called as a primer annealing. Is it clear? Yes, Yogita. So PCR we have discussed. So here PCR uses 2 sets of the primers. There are complementary regions, different varieties of primers are there. Then DNA polymerase enzyme, this will add the nucleotides. So we discussed the Thermus aquaticus. This is the temperature resistant one, and you did not stop the PCR mission for every cycle after. This is a revolutionary invention, Thermus aquaticus and its DNA polymerase. That polymerase is what is called as Taq polymerase. We discussed these things. Then 4 varieties of nucleotides will be added, and we have also compared the DNA replication with this PCR, if you remember correctly. The DNA replication is in relation to the mitosis, right? So, and the amplified one can be ligated with the vector for further cloning. You have to now use this ligase enzyme, to combine this vector DNA and also this one. See the insertion of this recombinant DNA into the host. How to insert this recombinant DNA? And what about this, ligase action? Ligase action. Ligase action is it will ligate. The vector will be ligated with the source DNA. So now what you should do? Now recombinant DNA is ready and it should be introduced into the host. There are several methods to introduce. Okay. And in tomorrow's session, I will discuss these methods and we will also proceed further. And tomorrow we will try to finish this, and we will also have one exam on this. You please, mentally prepare for the examination. Right? Any questions here?
No, sir.
No, sir.
Yeah. Right. We'll meet tomorrow.
Okay, sir. Thank you, sir.
Okay, sir. Good evening, sir.